I'm changing my title somewhat to address the open questions in neuroscience, and it seemed to me worthwhile, even though this will be review for many of you, to ask directly the question of how good is the mouse cortex as a model for the human, because one of the projects at the Institute is to understand the human brain, and one of the means toward that is to study the mouse. And then I also want to talk about how does action affect perception and plasticity. We know it does in humans, and can we study its effects in the mouse? Well, there are a few prominent differences between mouse and human cortex. And actually, much of what we infer about human cortex we know from monkeys, who have very similar development, very similar psychophysical properties, and very similar anatomy to humans. And some of the prominent differences are that mice and other rodents lack orientation and ocular dominance columns, lack this clustering of response properties that's characteristic and that's been studied a lot in the large cortex. But there are many compelling similarities. There's the same basic structure and cell types as defined by many different criteria. There's a similar sequence of developmental events. There are multiple visual areas in the visual system. There are similar response properties and receptive fields of single neurons, and I'll talk about that in detail, and similar activity-dependent plasticity. But first, looking at what's different is these large animals do have columns, and if you look down on them and label them, you see in cats and monkeys, and also in humans, in this slide made by my colleague Jonathan Horton, this prominent clustering of input from serving the left and right eye into separate patches of cortex in the human visual cortex. And Dr. Horton's drawn this on the photograph of the brain of these people. But even Dr. Horton, who spent much of his career studying ocular dominance columns, in the end concedes that, as I have, in the end concedes that it's the pattern of connections onto individual neurons, it's the responses of those neurons, which is specifically important, and not the millimeter scale organization of these columns, because mice and the rodents lack them. But if you look at a sequence of events, which I've just drawn here in terms of topographic map refinement, elaboration of highly specific receptive fields, a period of plasticity that allows the matching of receptive fields in the two eyes, and then a period in adulthood of much less plasticity that allows much slower and smaller changes. These are similar in the mouse and in large animals and in the human from clinical data. So let's look at the individual cells in the primary visual cortex. In the work of Hubel and Wiesel, it was discovered that these cells were selective for stimulus orientation. This is the first simple cell that was plotted and appears in their first paper. And it's possible to study these things quantitatively with computer techniques. And here's a picture from one of Clay Reed's papers on the parametric simple cell, linear simple cell in the visual cortex. And Chris Neal in the lab studied these cells in the mouse using silicon probes with allowed recordings from many cells at once, and had important advantages in that it allowed one to distinguish the putative excitatory cells from the putative inhibitory cells with no overlap whatsoever because of the geometry of the recording setup. And it also allowed one to locate sites, locate the anatomical sites of the recording to different layers using current source density analysis, something that wasn't possible with physiological means. And using this approach, he plotted the receptive fields of the typical cell in the upper layers of mouse visual cortex. And those are typically linear simple cells that could have come out of Hubel and Wiesel's 1962 paper or their later papers on the monkey visual cortex.
If you change the scale bars.
If you change the scale bars. I mean, if you can't, right, if you change the scale bars in terms of degrees. And these cells are highly selective for orientation, like the ones in large animals. And indeed, the cells that are not selective for orientation in the upper layers are predominantly these putative inhibitory neurons. So in fact, practically all the excitatory neurons in the upper layers are selective for orientation. Most of these excitatory neurons are linear, and they're also nonlinear ones. So these correspond to the simple cells of Hubel and Wiesel and the complex cells of Hubel and Wiesel. And even in the mouse cortex, these neurons do very elaborate things, such as contrast invariant orientation tuning, which has been the subject of a great deal of modeling because it takes a sophisticated circuitry to produce this. Practically all the cells are responsive, most of them simple cells in layer 4 and the upper layers. And in a means of plotting receptive fields that's scale invariant that was devised by Dario Ringach, when Chris put the data for the mouse, plotted it on top of the data for cats and monkeys, it just fits in the very same distribution. So what it looks like is that the mouse sees the world very much the way the cat does, except for a given picture that he looks at, he has to be 20 times closer to see it because he has very poor spatial resolution. But the intrinsic properties of the cortex in analyzing it at the level of resolution that the animal has is very similar to what is present in large animals and to what we believe is present in the humans. So on that score, the mouse is very similar to the large animals. And it doesn't matter how you measure this. I messed up the movie, but you can measure it with microelectrodes, or Chris has measured it with calcium signals, and you get the very same properties from these cells. So another major way in which mouse visual cortex is similar to that of the large animals is in its plasticity properties. Rapid activity-dependent plasticity with a well-defined critical period was one of the critical features that Hubel and Wiesel discovered. The mouse has a binocular region of the visual cortex. Within that region, there is rapid activity-dependent plasticity that has a well-defined critical period. You can plot it with microelectrodes, or you can measure it behaviorally. And the mouse is also similar to these other animals in the relative amounts of plasticity in the different layers and in the rates of plasticity in the different layers. So in these respects, the mouse is very, very similar. But of course, the mouse has tremendous advantages that we can use a variety of techniques to track responses over time in individual animals. And when we did this, looking at the sequence of events during the induction of plasticity by occluding the vision of one eye briefly during early life, what we see is that the initial effect is a loss of response to the open eye, to the closed eye, and then that response stays stable after an initial very rapid loss. And then when you open the eye again, the response comes back very rapidly to its original state. Whereas if you look at the responses to the closed eye, during this initial loss, there's no change in the response to the closed eye. I'm sorry, there's no change in the responses to the open eye, but thereafter, the response to the open eye gets very much better. And then if you open the closed eye again, things go back to the initial state. So I don't have time to tell you about the series of genetic and chemical genetic techniques that have been applied to study this process in the mouse. You can divide this plasticity regime into 3 parts. And we have been able to isolate the signaling mechanisms that are involved in the plasticity of each of these phases of plasticity. And there are distinct mechanisms involved in each of these processes. And these lead to hypotheses about what changes in the circuitry are responsible for them. So now what I want to address is, how does action, how does doing things affect perception and plasticity? And can we study these effects in the mouse? we've studied this by making recordings from a mouse on a styrofoam ball that floats on air, a preparation that was devised in David Tank's lab and that they used to study the hippocampus to great advantage. And you can look at a mouse. As far as the mouse is concerned, he's walking on an infinite flat surface, and he can walk as much in any direction as he wants, and they're quite comfortable doing this. When Chris analyzed recordings that were made while we studied the receptive fields of neurons of animals on this trackball, what you could see is the very same properties that we see in anesthetized mice. So that was, in fact, a very important goal, to see whether the development and the plasticity of mice, which happens when they're walking around and not anesthetized, is similar to what we study in anesthetized animals, and it is. The basic properties of the neurons, as far as we can tell, are all the same. But strikingly, when the animal is walking or running, the responses in the visual cortex were much bigger, and we've outlined in blue the trials when the animal was walking, and in red when the animal was stationary. And what you can see is the selective properties of the neurons were the same, but the responses are much bigger. And this is a cortical property. It doesn't happen in the visual input to the cortex. There is not a similar change in the lateral geniculate nucleus. So this really turns out to be a dramatic activation of the cortex, which doesn't change spontaneous activity. It dramatically increases the response to an optimal stimulus without changing selectivity. So paying attention or action really has a very profound effect on the responses of neurons in the visual cortex. And it can be studied with microelectrodes or calcium imaging, and the result persists independent of techniques. Now, Bob Wirtz, who was the one who did the gold standard experiment in monkeys and first showed that the properties that Hubel and Wiesel had seen in anesthetized monkeys are present also in the alert animal, and presumably in each of us as we walk around and as we go to this lecture, wrote in his memoirs that the modulations that are associated with action and attention that he expected to find in the striate cortex really aren't there very much in the monkey, but they are found prominently and studied very prominently by John Mansell, for example, at higher cortical stages in the monkey. In the mouse, these modulations are large, as Chris's work has shown, even in V1. And perhaps this is a case of encephalization. And with fewer areas, the mice might have to do more in the earlier ones. And indeed, I regard this as a promising feature for the study of the mouse, that we can see things going on in association with important events in cortical function at an earlier stage in visual processing. But in fact, if you look at higher cortical areas in the monkey, the same thing is true. So Chris and we were interested in whether is it the locomotion itself or is it some ascending form of neuromodulation that enhances these cortical responses. And we took advantage of some work that was going on in Antonello Banci's and Linda Wilbrecht's lab, where in the pedunculopontine tegmental nucleus, they had discovered that activating those cells, which they could do optogenetically, made an animal walk. And the circuitry made an animal walk or run. And so what they devised after labeling the glutamatergic cells in this nucleus, some of which project forward to the anterior forebrain, and some of which project down to cause locomotion, is they could make the cells fire. And if you look at a mouse on the trackball in experiments that Moses, Lee, and Chris Neal did, you can see that every time you see the laser fire, the animal starts to walk on the trackball, and when the laser stops, the animal stops walking. So we hoped we could use this to investigate whether it's this ascending neuromodulation or the locomotory circuits themselves that are responsible for cortical modulation.
Is that direct a motor effect?
What?
Is that a direct motor effect?
It's a It's an integrated effect. It includes neuromodulation and locomotion. And so in anesthetized animals, the response enhancement was seen, and I'll summarize it by showing this picture of the EEG sign, the increased gamma oscillations which are present in anesthetized animals. But that didn't seem very informative because the animals, we might have vetoed locomotion between the locomotory circuits and the actual walking with the anesthesia. So if you stimulate with a laser at high rates, you reliably induce locomotion. But if you stimulate with the laser at lower rates, then you reliably induce locomotion, and you induce an increased response of the neurons, and you induce this gamma oscillation. But if you stimulate with the laser at lower rates, you don't induce locomotion, but you still induce the increased gamma oscillation, and you still induce the visual response gain. So it looks like it's this ascending neuromodulatory input which is being activated from the tegmentum that's important for the visual response gain. So the final question I want to talk about is this enhancement of responses by action, does it increase plasticity in the cortex? If you're getting 3 times the size of the cortical response that you were previously getting, does that make the cortex more plastic? And to analyze this, Megumi Kaneko in the laboratory studied the recovery from monocular visual deprivation in adults. And in normal adults, this recovery is very slow. And even after very long times, after weeks and weeks of vision with the eye open, it never reaches the normal range. And that's the clinical finding in humans who are recovering from amblyopia. It just doesn't work very well. And so what Megumi Kaneko did, was to give animals a pattern of visual stimulation while they were running on a trackball to see whether this increased activity would facilitate recovery. And she stimulated, the visual stimulation was either a visual noise pattern matched to the spatiotemporal frequency sensitivity of the neurons, or else it was drifting bars and gratings. And it's quite evident it's been measured a number of different ways, but looking with intrinsic signal imaging, you can see that the animals that received visual stimulation while they were running, the response to the closed eye recovered much better than it did in the control animals. And indeed, if you plot it quantitatively, within a week, the closed eye response almost reaches the normal range, which it doesn't in the control cases. And this recovery depended on the conjunction of the enhanced responses produced by running and the visual stimulation. So if you gave the same amount of visual stimulation while the animals were in their home cage, or if you gave the same amount of running without presenting the visual stimulation, you did not get recovery of function. And even more surprisingly, the recovery you got was stimulus-specific. So remember I said we gave experience either with a visual noise pattern, the blobby thing there, or with bars and gratings. And if you measured the response to the visual noise pattern after experience with the visual noise pattern, it recovered very dramatically. The response to bars and gratings did not recover. If you looked at the response to bars and gratings after experience with bars and gratings while they were running, it recovered very dramatically, but the response to the visual noise pattern didn't recover. And if you analyze the responses of the individual cells in the animals that recovered stimulated with bars and gratings, oriented contours, you recovered the ability of the cortex to match the preferred orientations in the 2 eyes. And it matches just about as well as it does in normal control animals that have never been deprived. And that's quite different from the result in the others. So finally, sort of what's next? It's important to study the development of the circuitry and the actual plasticity, either in the way that Clay Reed so elegantly described, or at lower levels of resolution. And we've taken advantage of the Allen Institute AI-14 mice to label neurons in work that Sebastian Espinosa did, and in the same neurons label either their postsynaptic densities by co-labeling the cells with PSD95, or labeling their presynaptic terminals, and then following these in 2-photon microscopy over days. And the analysis hasn't been complete yet, but in many instances, in none of the control instances, and in many experimental instances, we see really dramatic changes in a few days that are similar in time course and similar in magnitude to the changes we see in the physiology. So we think these changes in the circuitry are sculpting the changes in physiology. I just want to show you pictures. Actually, Chris has kind of cleaned up his act. He's sitting in the audience here. His lab is at the University of Oregon. Megumi Kaneko is still in my lab. And Chris did the work on the physiology of the visual cortex, and Megumi did the work analyzing the plasticity of the visual cortex. So thank you.